Bismillah Rahman Rahim. Welcome back, friends. Welcome to another video lecture by Kami Biology. In this video lecture, we are going to talk about nucleus structure, means eukaryotic nucleus structure. So let me write nucleus structure. Okay, so obviously, the nucleus is always present in eukaryotic cell, not in prokaryotic cell. Now, in the structure of eukaryotic nucleus, what we are going to talk about? We are going to talk about chromatin, we are in chromatin, especially we are going to talk about euchromatin, and heterochromatin. Then second, we are going to talk about nuclear envelope. Third, we are going to talk about the nuclear pores, nucleolus. Now these are the main topic of our lecture today. Okay, so before starting the structure of the eukaryotic nucleus, let me tell you the introduction of eukaryotic nucleus. You know, nucleus is a very important organelles that are usually present in a eukaryotic cell. We also call that what? We also call that heart of the cell. What we call that? We also call that heart of the cell. Heart or control center. Okay, so we also call it heart or control center of the cell. Why? Because it control all activity of the cell. Okay, you know, this nucleus was first of all discovered by Robert Brown in 1831. So let me write, it was discovered by whom? It was discovered by Robert Brown in 1831. Now nucleus, if we see the nucleus, so first of all the diameter of the nucleus, let's suppose this is the nucleus, so the diameter of each nucleus will be 5 to 7 nanometer. This is the diameter of the nucleus. Now let's talk about the structure. You know, as you are watching this green one area, this means inside the nucleus, these area are called chromatins. What we call that? We call that chromatins. We call that chromatin. Okay, chromatin are actually present in the nucleoplasm. This one area, this white one, as you are watching, this white one area are called nucleoplasm. Now what is nucleoplasm? Nucleoplasm are actually a fluid or a liquid that are usually present inside the nucleus. Okay, just like in cell, cytoplasm are the fluid. Similarly, in nucleus, the fluid are the nucleoplasm. Now let's talk about the chromatin. You know, chromatin further contain two main structure. You know, these are the chromatin, these green ones as you are watching, what these are the chromatin. But chromatin further contain two main structure. The first structure we call it euchromatin and the other we call that heterochromatin. And each chromatin are what? Each chromatin are the dense fibrous like material. Okay, that are usually appears when we stain the cell. Now one thing more about the chromatin that I must tell you, these chromatin are actually in a dense form. Okay, you know, all these chromatin are actually DNA containing. You know, if we talked about the DNA, inshallah I will make a separate video lecture on a DNA in a detail. Now the chromatin is you know, look like all the chromatin, you know, whenever we talk about the DNA, so actually the DNA are made up of this chromatin. Now whenever the cell are ready to divide, during non-dividing cell, these chromatin are what? In a dense form. But one thing more that I must tell you, whenever the cell are ready to divide, then what actually happen, the chromatin are condensed. They are condensed and they make a structure, visible structure, which we call that chromosomes. Okay, so chromosomes will be appear during a dividing cell. It will be not present usually, it will be appear during cell division. So then this one area which are usually present in the nucleus, we call that chromosomes. Chromosomes. You know, chromatin consists further two structure. The first we called it euchromatins. Now these black one, as you are watching, these black ones are representing what? These black ones are representing euchromatins. What? Euchromatin. Now euchromatins are those chromatins that are loosely connected to each other. They are loosely connected to each other, and they are those chromatin that can express the eukaryotic genes actively, that can express actively the eukaryotic genes. On the other hand, we have another chromatins, these green one as you are watching. These green one, we call that what? We call that heterochromatins. Okay, what we call it? We call that heterochromatin. Heterochromatin. Heterochromatin is opposite to this euchromatin. Heterochromatin are strong and tightly connected, and it is coiled. Okay, and when we see it under the microscope, so it will be a dark in shape, means dark in color. Okay, when we stain it. Now one thing more, that this chromatin are not usually expressed the genes, so it will be not present usually. It will be present during cell division. So this is about the euchromatin and heterochromatin. Now let's talk about another structure of the nucleus, that's about the nuclear envelope. Okay, this one area, means this outer area of the nucleus is called what? This outer area of the nucleus is called nuclear envelope. This one, what we call it? We call that nuclear envelope. Okay, nuclear envelope is a layer that are surrounded by a nucleus. Okay, or we can say it is a membrane of the nucleus. Okay, due to which the nucleus are covered. Nuclear envelope or nuclear membrane, there are two membrane. Okay, the one we call it outer membrane and the other we call it inner membrane. As you are watching this one, what? This is the outer, let's suppose, outer nuclear envelope, and this is what? This is the inner. Okay, there is a space between outer and inner. As you are watching this one space, we call this space what? We call that pre-nuclear space. So we can also say that outer and inner membrane are separated with each other by this pre-nuclear space. Okay, and the separation of outer and inner membrane will be almost, if we see the length between them, so it will be almost 15 to 70 nanometer. Now each nuclear envelope, means the outer envelope of the nuclear envelope, is usually connected with endoplasmic reticulum. Okay, now we have inside the animal cell we have a lamina. Okay, lamina. So lamina are usually present in this inner membrane area. And one thing more, lamina are actually, it is present on the inner membrane surface. Okay, and it usually present in case of animal cells. And the lamina are usually used to support the nuclear envelope inside the animal cell. Now one thing more, if we talked about the inner membrane, so as you are watching, these chromatin material are usually connected with what? These chromatin material are usually connected with the inner membrane, not with the outer membrane. Okay, so now our next structure of the nucleus is about this nuclear pores. Nucleus also contain a small holes, as you are watching these red one. Okay, these red one are what? These red one are the nuclear pores. Nuclear. So the diameter of nuclear pore will be 70 nanometer. Now nuclear pore is used to transport different material inside the nucleus. Okay, one thing more, the ribosomes, which are specially synthesized by this nucleolus, these ribosomes are then released out through this nuclear pores. Not only the ribosomes will be released out, DNA which are present inside the nucleus, whenever it is ready to make ribosomal RNA, or whenever it is ready to make messenger RNA, so then messenger RNA or ribosomal RNA, whenever it is synthesized inside the nucleus, then each material are also released out by this nuclear pore. Sometimes the material are also internalized to this nucleus. If you remember in HIV viruses, the HIV genetic material usually it get internalized through this nuclear pores, and it is connected with the genetic material of the eukaryotic cell. Okay, so it is not only used that the material which is synthesized inside the nucleus will be released out through this pore. Now all those material which are present outside the cell, it may be internalized to this pore as well. Now if we see all these pores, so it can occupy almost all these pores can occupy 10 to 20% of the nucleus surface. Okay, so it can occupy 10 to 20% of the total nucleus. Okay, one thing more about the nucleolus, there are another structure which are also present at the edge of the nucleus, at the edge of the nuclear pore, as you are watching. Now these structure are called, what we call this structure, annulus. What we call it? We call that annulus. So let me write the name, annulus. Now annulus are used to strong the pores or to strong the nuclear holes. Okay, so that's all about the nuclear pores. Now let's talk about the nucleolus. This one portion, as you are watching, this dark portion, we call that what? We call that nucleolus. So nucleolus is actually the central part of the nucleus, and nucleolus, the main action or mechanism of the nucleolus is that they can synthesize the ribosomes. Once they synthesize the ribosomes, then the ribosomes release out from each nuclear pores. So I make here one nuclei, but many of the cell contain we have some of the cell that contain 2 nuclei, some of the cell can contain 3 nuclei, nucleoli. Okay, but one thing more, this nucleolus doesn't contain what? This nucleolus doesn't contain a membrane. Okay, as nucleus contain a membrane, but nucleolus doesn't contain a membrane. And some cells have 2 nucleolus, some cells have 3 nucleolus. So it doesn't mean that cells have only 1 nucleolus. Okay, one thing more that I must tell you about the nucleolus features. Nucleolus are always present in non-dividing cells. This is very important point. Nucleolus are always present in non-dividing cell, but whenever the cell are ready to divide, then this nucleolus will be disappear. So you can't see the nucleolus during cell division. But yes, one thing more, when the cell are formed, then you can easily see these nucleolus are reformed again. Okay, one thing more, these nucleolus are also responsible to synthesize the ribosomal RNA as well. Why? Because if we see, these nucleolus can connect it with the DNA as well. Okay, so that's why we can say it is also responsible to make what? To make the ribosomal RNA as well. So that's all about the structure of the nucleus. I hope you understand about this video lecture. If you like the video, so make sure to hit the like button, share this video, and don't forget to subscribe the channel. Subscribe the channel to get more videos like that. Thank you.